So this is HIV. It's a typical retrovirus, meaning that it has an outer envelope, and in the center it has 2 copies of RNA, as well as an enzyme here in blue that's reverse transcriptase, which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein. What ultimately happens is fusion of those 2 membranes, and the viral genetic material is injected essentially into the cell, and the envelope protein is left at the cell surface. The virus has a matrix and a capsid protein, shown here in green and red, that essentially are digested when it enters into the cell. That releases viral enzymes and the viral RNA. And here we have reverse transcriptase, which takes the viral RNA and, using host nucleotides, converts that viral RNA into a single strand of DNA. Stranded DNA now is again reverse transcribed into a double-stranded DNA. At that point, another enzyme that has come in with the virus in the beginning, called integrase, essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and it basically, the integrase enzyme, makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum. Mature structures that make up the final virion, and now you have a mature infectious virion that can go on now to infect other cells. Once that happens, now the cell can produce tons of viruses, and this is really what then keeps the whole process going.